Hey guys, so the science video that I promised you guys, thank you for tuning in. As many of you know, this is going to be my very last video. So as some of you guys know, if you've been staying up with some of the vlogs that Amanda and I did, I was working in a research lab these past 8 months. And I had the opportunity, thanks to my supervisor, to be able to train a few people, some new graduate students and some undergraduate students. And I had the opportunity to build my passion of teaching, and this brought me to you guys again today. So stay tuned for my new video. So a few of the things that I did in my lab was I had the opportunity to train my students how to do a technique known as Western blotting. So what Western blotting is a way of detecting protein levels in a particular treatment by running samples on a polyacrylamide gel and then running a current through it and then transferring it onto a piece of paper known as a membrane. So the way that this works is, to begin with other stuff, there's two typical types of gels that you would use for something known as electrophoresis. Electrophoresis is the method by which you use a current or a voltage to separate DNA or protein and sometimes RNA based off of molecular weight for further testing. And this is very useful for numerous things. For example, forensics is a huge thing when they do DNA fingerprinting or when they do protein assays. So the first thing that you would do is you would have protein lysates that you would have to boil down to denature any proteins that would be there. And then you would, from these two gels, you would either use an agarose gel, which is used for DNA and RNA probing, and that's run vertically, or not vertically, horizontally, sorry. And then you would have protein samples that you would run on a polyacrylamide gel that contains a detergent known as SDS, or sodium dodecyl sulfate. And that gel would run vertically. And you would load a molecular weight ladder that would allow you to figure out where your protein will be based off its molecular weight for when you go to probe. So the way that this works is when you run your gel, you would then take that gel and you would run a current through it once again, so that all your protein separates onto a membrane, which is a special type of piece of paper. There's two types once again. There's a PPDF, which is polyvinyl difluoride, and the other one's a nitrocellulose membrane. Typically I use a nitrocellulose membrane, and you would transfer those through a medium, and then you would begin your probing. So the first thing you would do once your proteins are on your membrane is you would block. And typically what blocking means is you add milk to your membrane and let it shake at a moderate speed for an hour, sometimes less, sometimes more, based off of the type of protein that you're probing for. And what this does is the milk proteins actually bind to different parts of your membrane so that all the non-specific areas on your membrane are then blocked for when you would add a primary and secondary antibody to bind to the protein of interest. So after you're done blocking, you would add your primary and either incubate it for a couple of hours at room temperature, or you would incubate it overnight at 4 degrees. What this antibody is a small molecule that has two ends that are typically specific to the proteins on your membrane, which are known as your antigens, and they would bind to them. And then sometimes you could have them labeled with a fluorescent marker, which would usually be used for something like ELISA, which is another immuno technique, in which it's known as direct ELISA. Typically in Western blots you don't have something like that, but you can if you know that your protein levels are very high. And then you have something known as a secondary antibody that has this fluorescent marker. And in between the two, you would do numerous washes just to remove any kind of non-specific binding by the primary. And then you would add your secondary, and your secondary would attach to your primary antibody. And then you can add a substrate. Typically for Western blotting, it's ECL, which is a chemiluminescent substrate, which then would allow your membrane to fluoresce in that particular area when you were to take it to the dark and expose it on film. So, one of the questions that one of my students asked me was, why is it that when we wash our membrane after probing with our primary and secondary antibody, why does the detergent not remove the primary and secondary antibodies from your protein of interest? Why do they remain? So that's actually a very good question, and it's a very legitimate question, and I totally understand that question. And the reason is that when your antibodies attach non-specifically anywhere to your membrane, their link is not strong enough. So the way that I described it to them is that, say for example, we're linked together. Amanda, I'm going to call you over here for this demonstration.
Oh goodness.
Okay. So say for example that Amanda and I are holding fingers, okay? And say I tried to pull away. This is the non-specific antibodies. So I would pull away, and it's very easy to break away, okay? So when I have my detergent, it's going to go in between the two, and it's going to wash away any of my antibody that's attached somewhere else. But now, if I link arms and someone were to pull me apart, I would not break. Because now, this is when it's specifically bound, and now you have covalent bonds or disulfide bonds bridging the two antigen and the antibody together, so that when you go to wash it, the bond is too strong for the weak detergent to break it off. So that's the answer to that question.
Yeah.
The second question that they asked me, which I will thank one of my professors for answering this question for me when I was actually taking immuno, but the second question was, now why do you require a secondary antibody for a Western blot and sometimes not for ELISA? And sometimes you do need it for ELISA and other immunotechniques. Why is the secondary required? So thanks to my professor, he did answer this question for me. And the reason behind this is when you have one primary antibody, okay, say you have your membrane that has your antigens on it, and you add your primary. One of your primaries will attach to one antigen, okay? Then for that primary, when you add a secondary, the secondary is typically less specific than the primary, so numerous secondary antibody molecules will attach to that one primary, and therefore it would cause your signal to be amplified, which is why I mentioned that if your protein levels are high, you don't typically need a secondary antibody, and you can use a primary that's labeled, but that's in certain conditions. Typically for a Western blot, there's only so much protein that you can load, therefore you do require a secondary antibody to amplify that signal so it's visible. So the answer to that question is it's there to amplify your signal. And that ends the process of a Western blot and some of the theory behind it. And one of the other things is that, so the reason why they use Western blotting, first of all, is to see, like I mentioned earlier, that you want to see the effects of your treatment on a certain thing. And sometimes it's just easier to run a Western blot because, for example, you don't get the result the first time. You can always remove your previous primary and secondary antibodies and reprobe for a different protein to see if that gives you something. And typically you would need something like a loading control at the end of it to make sure that all the information that you got is not because you loaded your lysates incorrectly, but because your treatment actually had an effect. So with that, I wrap this video, and I look forward to talking to you guys soon, and hopefully I can still continue vlogging. And I'm sorry that this is my last science video, but with school starting today, it's gonna get really hectic. But wish me luck, and I wish you guys all the best. Bye.